portfolio of over 200,000 analytical methods, Eurofins provides a unique range of analytical testing services to the pharmaceutical, food, environmental, and consumer products industries and to governments. Before we begin the presentations, we're going to pause for a quick polling question. And the questions should now be on your screen. Select from one of the answer choices. I'll leave the poll open for about 30 seconds before I close it and share the anonymous results. And the question is, do you currently use molecular markers for hemp? Okay, I'm going to close the poll now. So it looks like the majority of you do not, which is fine. And I will pass on the presentation to Asgar Shir, and he will let you know more about molecular markers applications for hemp.
Hello, everyone. My name is Asgar Shir. I am operation manager in Eurofins BioDiagnostics. A little of my background: I've been with Eurofins 11 years, managing a genetic lab here in Colorado. And before that, I was with a seed company and doing molecular breeding for that seed company. My training is plant breeding and genetics, and today I'm going to talk about application of molecular markers in general in plant breeding and production, as well as hemp breeding and production. So the first slide here, I'm showing why DNA markers and what are the advantages of these markers. Why do we want to use DNA markers? The first point is fast results. We can have results very quickly. In the case of hemp, for example, we have molecular markers for sexing that we can detect females from males, and that could be done as fast as 3 days. We have a lot of other examples in different crops, but here for hemp, specifically related to hemp, this is a good one. This type of testing is precise because it's at DNA level. So what you get is actual genotype that plant or that individual sample has. We don't need to have large number of samples like whole plant or anything like that. We just need a leaf punch if we are testing a plant, or we can do it at seed level actually also. So very small quantity of sample is required for this testing, which is a huge advantage. And the last one, which is very directly related to what you do as growers, breeders, processors, is how do you make sure that the material that you have is the correct material? And this one is when you want to test something and make sure you have, for example, in case of female, you have 100% feminized seed, or if you want to make sure that something that you bought actually is something that you received. And we have numerous number of projects that we've done in different crops for this type of work. One of the most important application of molecular markers is database application, and that is when we create a DNA fingerprint between your materials, basically, for your materials, for your germplasm, and that would show how these materials genetically are related to each other. Are they close sisters? Are they different lines? The same type of data can be used then for intellectual properties. If we want to patent something, we can use fingerprinting, DNA fingerprinting for that purpose. Also make sure that your lines, strains are protected. And a lot of time, again, when you have a good understanding of the material relationship at the genetic level, selecting parents for crosses that you are making makes more sense, because you can gain a lot more from your selection breeding for materials that are not too close to each other. Sometimes if we cross materials that they're maybe sister lines, the genetic gain is not very much, and we want to know how these materials are related. A good example is corn, actually, when there are 13 families, and when you go between families, you get the maximum of genetic gain. Did I pass on? No. Okay. So as I mentioned before, materials that we can use could be anything, type of material that we can use. It could be inbred lines, it could be strains, varieties, cultivars, hybrids, populations. A different population could be used for this type of testing. Then we can do the same thing on seed, leaf, root, any part of the plants. And the number of plants could be to test could be individual samples, and in some cases it could be bulk of samples, bulks of 5 different plants, 10, and even more than 10. So any type of material we can use for this. I'm going to just talk about indirect selection. So when we are using molecular markers, basically we are doing selection indirectly. And the principle here is very simple. You have a gene, we have a gene, and we don't want to wait for the gene to be expressed, so we try to use discover and find a molecular markers closely associated with that gene, and then use that molecular marker for the selection of the gene. Here as an example, in hemp is that we have hemp, we have female and male plants, so we have X chromosome and Y chromosome. And if we have a molecular markers that it can actually differentiate between X and Y chromosome, then we can determine which, each plant, is it a female or male? And that is indirect selection for female, male. And with this, we don't have to wait for the plants to flower actually and see it. We can do it at very early stage on the plant, or if we can even do that one on the seed. And I'm going to talk about feminized seed in a couple of minutes and show you how we do it right in our labs. Here is another example of indirect selection. This is disease resistance. It could be, in this case, is in tomato, but it could be, this is actual data from our own lab. It could be anything that is also in hemp. If we work on a disease-resistant that is important in hemp, and we can develop this type of molecular markers and technology. 3 different technology is shown here. I'm not going to go through the whole each one of these in details, but basically what we see here, the standard, classical way is the agarose gel, and in that case you have a band for resistant, a band maybe for susceptible, and then the progeny between these 2 parents is going to be showing the both bands. Same thing here, 1 band for resistant, 1 band for susceptible, and progeny which is resistant, it shows this both. And in this one, which is a SNP technology, is basically the same thing, resistant lines, susceptible, and then you have those that are progenies of resistant and susceptible. As I said, this could be used for hemp or any other crop. Advantages that we gain from marker-assisted breeding, the first one is the speed, because we can select best plants that they have the gene and then advance those to next generation. That could save us generations. It could save us space, the next one, which basically we can select those plants that are, if we are working with resistant, or if we are transplanting samples from greenhouse to field, or in the greenhouse, we can only select those that we want to move them to field or move them to next generation. So that would be space saving that we can gain from this. Timing is one of the advantages, because if the trait is expressed very late in the season, we don't have to wait until that time. We can actually look at and detect those resistant lines at the very earliest stages. Another advantage is stacking many genes, because with the molecular markers, one DNA extraction, we can test for many different traits, which is usually not possible if we go the usual phenotyping. And of course, efficiency, in case, when we are using molecular markers for backcrossing, instead of going 5, 6 generations to get the pure line plus the gene of interest, normally we can do this in 1 or 2 generations by just using background markers for our for our line of interest. And the example that I was talking earlier about hemp, this is a gender marker that we use in our labs. The marker can show which plant or which sample is female and which sample is male. In this case, we use 96 feminized seed. We can also do it on the plants, but most people at this point, they're producing seed and they want to make sure their seed is feminized 100% or whatever percent that they desire. And then so 96 seed, we run it. We include our controls in the lab. We have a female and male control that we use. And in the end, we generate this type of certificate that it shows, customer information, all of our own information, how many plant or seed we used, what was the number of samples that reported, percentage of female, percentage of male, and that would go back to customer as results and as a certificate to show their lot and percentage of female in their lot. Another type of work that I already talked about earlier is, genetic relationship, DNA fingerprinting of materials, germplasm of customers. In this case, we have a panel of 18 hemp markers. These are specific markers that they have shown power of discriminating between hemp strains, and any number of lines, any number of entries that it comes from customer. In this case, for example, this is a project that we recently did. We had 23 entries and 18 markers, our standard panel of markers. And the results was clearly, we got two groups here, A group and B group. And when we look at the genetic configuration of this population, we see that one of the strains or lines that we tested is actually very different from, has a lot different between that one and everything else. On this horizontal axis is the percentage of differences between these lines, and that's how we can interpret this genetic tree, or the technical term is dendrogram. So once we do this type of project, we report a table like this. For every single entry, we have a number, which is basically fragment or band that we saw for that one for a specific marker. And then we put the data together and we report a genetic tree like this. And that one shows how these materials are related to each other. And I think that was my last one. Thank you.
Excellent. Okay, so now Anthony, you can start your presentation.
Awesome. Thanks, Asgar, and thanks, Genevieve. As Genevieve mentioned, my name, as the screen shows, my name is Anthony Lai. I'm the commercial manager for the Eurofins QTA lab in Cincinnati, Ohio. As it relates to the hemp industry, we provide rapid turnaround cannabinoid analysis services that are most commonly leveraged during the growing season and leading up to harvest. Now, in the next section, Garrett will be talking, in more depth about full cannabinoid profile testing that his labs perform by HPLC. Our analysis, and through which these observations that I'm going to share with you were made, is abbreviated, and it's focused on just the CBDA, CBD, as well as THCA and Delta-9 THC parameters. I'm going to be sharing information on a few interesting observations that we've made regarding field sampling for cannabinoid analysis, as well as some things to keep in mind to get the most meaningful data out of your samples. An important note before I get kind of any further into this section is that the information that I'm going to be sharing is not related to compliance testing or any of the specific instructions outlined by your local state Department of Agriculture for pre-harvest compliance testing. This is focused on practical, routine quality testing that you may be performing to make harvest decisions and understand what your yield is going to be based on how you're going to be processing this material after harvest. For some additional background, we worked with over 230 growers this season to provide rapid cannabinoid analysis. Throughout the 2019 season, we received and analyzed a few thousand samples. And it's from these samples and these analyses that the observations were made. I'll start out by laying out some of what we saw and then talk a little bit more about what that could mean for sample selection and sample composition. Okay, not all flowers are created equal. This sounds pretty obvious, and the variability of cannabinoid production flower to flower, plant to plant, even within the same variety and location, is widely accepted as a matter of fact. And it's something that everyone deals with and has to manage. From the third-party analysis side, we weren't necessarily made aware of all the possible variables, stresses, or growing conditions for each of the individual samples we received. However, in many cases, we did receive information related to the location on the plant that flower or material was sampled from. What we observed confirmed that the further down the plant you go, or the further away from the canopy in direct sunlight, the slower the cannabinoid production. Nothing earth-shattering there, and for the most part, this was expected. What was surprising was the magnitude of the drop-off. For example, if the top of the flower was producing CBDA in the 10% range, we were seeing a moderate reduction in the middle flowers, somewhere between 5 and 15% lower relative. And again, no surprise there, but in the lower flowers, we were seeing a pretty wide range of cannabinoid production performance. We were seeing anywhere from a 30 to 60% reduction in CBDA production when compared to the top flower of the same plant. So once we started observing this trend and discussing it with the growers that were sending us samples, we were able to get a little bit more information from them, as most were just as interested, if not more interested, in the trending we were seeing. This slide kind of illustrates just one example where we were able to have 4 different plants sampled for top flower, middle flower, and bottom flowers. And the trend in the drop-off, visually here is pretty consistent. However, we did notice that plants 3 and 4 had a flatter trend. It had less of a drop-off of CBDA and more consistent cannabinoid production across all of the flowers on the plant. When we started digging a little deeper, we found that plants 1 and 2 came from fields that were open, sprayed fields. But plants 3 and 4 came from irrigated fields where plastic sheet mulch was used. Now, again, this is just one example that illustrates something that we observed on a number of occasions, but by no means would these observations satisfy kind of the rigors of an official field study. However, these types of results were observed on a number of occasions throughout the season and across different growers. Getting the best return on your sample. So how does that information translate to field sampling? All of that information is important if for no other reason than to reinforce the need for composite sampling and sample homogenizing when trying to stay on top of the overall cannabinoid production across your fields. It's critical to represent material from multiple plants and flowers from all the different levels of the plant, as well as different areas across a plot, especially when there could be significant differences in growing conditions. A simple quadrant approach was widely adopted across the growers that were sending us samples with some variation. And again, they were collecting top, middle, bottom flowers from different plants within a quadrant and repeating that for each quadrant. So they were covering the edges to the center and again, different plants, top, middle, and bottom. Composite samples that we would receive would often come in 1-gallon bags and consist of 4 top flowers, 4 to 8 middle flowers, and a range of smaller bottom flowers. The sample volume and number of total samples was completely scaled to the size of the farm, the number of acres, and varieties that a grower would have. How will this material be processed? This is another critical question to consider before collecting and sending off a sample. What is this material going to be used for? How am I planning on selling it? Is this going to be bulk biomass, or will this be dried and the floral material stripped off the stalk? Or could we be looking at material being processed for smokable flour? Regardless of what the material will be used for, you'll want to match your sample selection and sample preparation with what it's being grown for and what that post-harvest processing will look like to get the most consistent results, both pre and post-harvest. We've received large sections of plants as a single sample. And because our test method is non-destructive, doesn't require the use of any solvents or reagents, we can easily reprocess, blend, and reanalyze samples a number of times without compromising them. Through our analysis, we've dried a sample, processed the floral material separate from the stem and stalk and leaf material, and then after analysis, blended them back together so that we could compare the impact on the CBDA concentrations and give additional information back to the grower. This gave our growers a better look and a better idea of what their CBD and THC concentrations looked like with different post-harvest processing and separation. Generally, we were seeing an impact to the CBDA concentrations in the range of 10 to 15, sometimes 20%. But if nothing else became apparent, no two samples were the same. So when you're really looking to understand what you have in the field, composite sampling and matching your sample prep to what the material will be used for are both critical ways to get more representative samples and more valuable, useful data from the samples that you are testing or possibly sending out to maybe Garrett's lab. And that is just a brief rundown of some of the observations that we saw, the effect of sampling, and the need for composite sampling.
Thank you, Anthony. Garrett, you can begin your presentation.
All right, sounds good. All right, thanks, Anthony. Good morning, everybody. My name is Garrett Zielinski. I am an analytical service manager for Eurofins. I work mainly with dietary supplement and hemp companies to help set up their testing programs. And within these programs, it's testing for ingredients and processed materials and finished products. So we can kind of help through the whole supply chain. Today, I'll be talking a little bit about, testing to, ways to use testing to ensure product and ingredient quality. Within the hemp CBD space today, there aren't any really good federal guidelines in regards to testing. So what a lot of people do and what we suggest is to use kind of relevant, similar FDA, USDA regs as a guide. It really comes down to having specifications for your process and for your ingredients or finished products and ensuring those are met. But there is a few different regs that you can use to kind of base your specs and quality systems off of. So one is 21 CFR 111. So that goes into the dietary supplements, and since a lot of the finished products are going to be labeled as supplements, this is a good one to use. If you're using hemp, CBD, and food products that aren't labeled as supplements, you can also look at the human food regulations, then also the animal food regulations. So you want to look at what your finished product is, whether if you're producing an ingredient or a finished good, and see kind of where it fits best and base your quality systems on appropriate regulations. Again, this is kind of a gray area, so it's not extremely straightforward, which makes it challenging for the industry, including testing labs. But as long as you're, ensuring that products are what they should be and don't contain things that they shouldn't, that's really the best place to start. So testing is a big piece of that to ensure, you have quality products and ingredients. So one of the important things to consider with testing is you need to ensure methods are fit for purpose, and that there's some type of, validation or data to show that the methods are working appropriately. So one of the big methods that industry utilizes is that for cannabinoid potency. There is an industry organization, AOAC, that has kind of method guidelines for certain methods. And this helps, laboratories, either external labs or internal labs, ensure that the methods they're using are appropriate. The cannabinoid potency is good for, the good cannabinoids, but then also ensuring that your THC levels are below any threshold. Additional testing is for terpenes. So those are the other kind of good compounds in hemp. And in addition to testing for the good compounds, you also want to make sure that your ingredients and products don't contain things that they shouldn't. So that's, pesticides, heavy metals, microbiological contamination, solvents, mycotoxins. And then also it's important to consider stability. So within the supplement industry, there are specific regulations that your products must have 100% of label claim at the end of shelf life. I think, best practice is if you're labeling a CBD or hemp product with a supplement facts panel is to have those same stability studies and go through that same rigor to ensure that you're meeting label claim. So the cannabinoid profile, the most widely used approach is to use an AOAC approved method. There are a few methods that have been approved by AOAC. These are fit for purpose for the ingredients and products that the methods are that are listed in the methods. So those methods may not necessarily be fit for purpose for finished products, such as gummies, liquids, and other things like that. So if you are testing those other ingredients or products, it's good to have some type of validation data to show that the methods are fit for purpose. And there's a few different ways to go about that with accuracy precision studies. But taking a method, an AOAC approved method that's approved for dried plant material, and just running that on a finished product isn't really ideal because you don't know if that method works well or not. So these are the kind of the main cannabinoids in the profile. The main ones that you'll find are the CBD and CBDA, then also the other kind of most important one currently is the THC. So terpenes, these are becoming more important as people look for the other beneficial compounds. So these kind of give products flavor and aromatics. There's a few methods out there for terpenes. Most of them are grass chromatography related. Here are just the kind of important terpenes for hemp. Some of these methods for terpenes will include a wider variety of other terpenes that can occur in other products, but these are the most important ones in hemp. And if you're looking for some of the beneficial characteristics, these are going to be the ones you wanna look at. Again, product safety is especially important with hemp, as we know there's a phytoremediator, so it'll pick up things out of the soil. You want to make sure that your pesticide residue, heavy metals, and mycotoxins are at an acceptable level before you further process the material, even harvest the material. In addition, residual solvents. If you're doing any type of solvent extraction, it's best practice to test your in-process materials and finished products for solvents. That would include any solvents that you use for the processing. Then also, in addition, you want to consider if any of the solvents you're using, if you're using ethanol, for example, if the ethanol you're using has any contaminants in it, any other solvents. Because we've seen, some toluene and things like that might be a contaminant in a solvent actually come through and not get taken care of with the processing and the cleanup of the material. So you don't just have to look at what you're using, you have to look at all the constituents of that and potentially test for any contaminants that might be in there that might come through. Microbiology, that's another big one. That can be challenging as well, since there's a few different approaches. You can either use the USP methods, which are more suited for ingredients, I think, and supplements. You can also use the AOAC methods, which is generally for food type products that are labeled as food. So here's kind of one example of why I should test for contaminants. So there are regulations out there about adulterated food and then also adulterated supplements. So here's an example. For foods, it says pesticide residue greater than specified limit makes a food adulterated per the regulation. So if you have pesticides above the level in your food, it's adulterated food and it's an illegal product. Same holds true for dietary supplements. The SHE has a regulation saying manufacturers, distributors are prohibited from marketing products that are adulterated or misbranded. So if the product has any contaminants in it, that product is considered adulterated, misbranded, and FDA could take action to get those off the market. Some additional testing considerations. So one thing that I think is particularly important with hemp is to test throughout the supply chain. It is a very expensive product, to harvest and to sell. So it's good to test, before you even plant the material, to test the soil to make sure that it, doesn't have any pesticides or heavy metals, or that they're acceptable levels, because the hemp will take those up. You don't want to plant something and then find out after it's already grown that it can't be harvested. So again, before harvest is a good time to test. Make sure that the product that your hemp has, adequate quantities of the beneficial compounds, and also has appropriate quantities of THC. You don't want to go through the cost and trouble of harvesting and processing if the material is not going to be good in the end. And again, after harvest, so after you've harvested all the material and processed it, it's good to test. You don't want to go through any type of additional processing or extraction if the material is not going to be good in the end. And then again, after processing, you want to make sure that whatever you're delivering as your finished product, whether it's an ingredient or a true finished product, that it is what it should be. It contains everything you want it to, and doesn't contain things that it shouldn't. And another thing to consider is, what is the intended use of your finished product? Is your product going to be an ingredient that goes into the food chain? Is it going to be a dietary ingredient that goes into a supplement? Or are you making a finished product, a finished food product, or finished supplement? And consider what specifications are going to be needed at that point. You really want to have this understanding before you even, start planting, in my opinion, to know what your end goal is. You don't want to have, a lot of material, finished products, ingredients, and not have a good way to sell them. So that's one thing that I've kind of seen a little bit. So it's important to know what you're going to do to ensure that you're doing the right process from the beginning. You can definitely save a lot of time and effort that way. And since the industry is still fairly new and there's a lot of gray area, and FDA hasn't really had too much concrete to say, it's good to look for guidance at different places that you can. So there are a few industry associations that have some great resources, and all these industry associations are really like their intent and their goal is to make sure that they're helping industry create quality, good products for the consumer. So these industries really have the consumer as the most important aspect, and then also how to help the companies kind of get there and develop products that do meet certain guidelines and are quality. So U.S. Hemp Roundtable, U.S. Hemp Authority certified. They have a very good program. Hemp Industries Association, UNPA and APA. UNPA and APA each have cannabis committees that are, the industry is involved in to try to help set standards and guidances to, help its industry helping industry do the right thing and kind of show FDA that, we are all on the same page. We want to create quality products, and we're willing to, go through the due diligence throughout the whole process to make sure that we are having a quality, safe product in the end. So that was just a little rundown of the analytical perspective. So I think now we will take any questions if any came through for Genevieve.
Thank you, Garrett. Okay, so let's start out with a question I think will be for Asgar. And that question is, what traits are of the highest importance for developing molecular markers? Asgar, are you there?
Oh, yeah. Sorry, I was on mute. My phone was on mute. So, so I think, this is a question that we usually like to hear from our clients and customers. I've been talking to many people, and there are several this type of traits that is of interest. CBD, CBG, all of CBC even he has mentioned to me, and there are some maybe resistant genes that would be of interest, and those are the ones that are in the pipeline for us to come up with molecular markers that we can help the industry and people that are breeding or using this producing hemp, and they like to use these kind of technology to speed up their programs. But as I said, would be interesting to hear from the audience also that what traits are of their is important to them also.
Thanks, Asgar. Okay, our next question I think will also probably be for you. How many molecular markers do you recommend for genetic fingerprinting of hemp lines? Well,
at this point, we have 18 markers that we would say that is doing what is, it should do. But we are also working on a larger number of markers with thousands virtually of SNP markers to have a better fingerprint of different strains. But right now, what we see, and these 18 can give us a general picture of what's going on. And in near future, hopefully very soon, we would have a lot more that we can actually go to a lot more detail for that type. And that is, as I mentioned, is in the magnitude of tens of thousands of markers.
Thank you. Okay, another question. Do you germinate the feminized seeds you test for percentage of feminization, or are you literally utilizing seed tissue to test for gender?
We do seed directly. We extract DNA directly from seed, and we go from there. Now, if they want to test plants, we recommend that they send us leaf punches. We don't need a lot of, as I mentioned before, we don't need a lot of materials. We just need 1 or 2 leaf punches of plant. But when we do actual seed testing, is directly from seed. And that's the reason we can do it so quickly in 3 days, or if we want to, of course, there is always lots of sample in the lab, but the test by itself, it doesn't take more than 24 hours. But we guarantee 3 days as one of our services.
Thank you. Okay, next question. I think this is for Garrett. Do you recommend stability studies for wholesale extract?
If the extract will be, on the shelf and sold to consumers, I would say yes. I think also, even if that, extract is an ingredient going into being sold to others who will further, formulate it, you would still want some type of stability data on your extract to show your, customers that it is as potent as it should be, and just so you know how long you can, hold on to it before it, degrades as to where you couldn't use it.
Okay, thank you. Can you talk a little bit about the accuracy of residuals testing?
Sure. So residuals, if we're looking at residual solvents, pesticides, aflatoxins, I would say, in general, the lower you go, the less accurate the methods are. Again, we're talking for pesticides, we're talking in the, parts per million, parts per billion range. So, methods at that level are going to be less accurate than something that's at the percentage range. What labs should be doing for their methods is looking at the AOAC guidance for method validation, and that actually calls out accuracy requirements, and it's level dependent. I don't know the specifics off the top of my head, but for like something at 1 parts per billion, the accuracy of a method might have to be 70 to 130% for like recovery, but at something that's 1% of your active, the accuracy should be 96 to 104. So again, lower you go, the less accurate methods are going to be. But also if you're looking at residuals, your specification should be higher than what the method limit of quantitation is. So if you have an LOQ of 5 parts per billion for a pesticide, your specification should probably be a bit above of that to account for any type of method variation.
Thank you, Garrett. We have another question. Is it true that the extraction process, whether ethanol, hydrocarbons, or CO2, removes microbials?
I'm personally not familiar with the extraction process and how that works with microbials. I think, intuitively it would make sense if you're using, ethanol, and ethanol kills microbes, that it would, kill anything that's in there. I think there's also the potential for introducing microbes, like the more you process something from either, personnel or equipment or processing. So there's kind of a few different, considerations you have to look at depending on what your process is. And then that's also a good reason to do that additional testing for your, process validation, to show that, if you are doing an ethanol extraction, that, you don't have microbes at the end, but then also to ensure that you have appropriate, recovery of your cannabinoids and other compounds.
Right, thank you. Okay, I think we're going to ask one more question. Everyone that's still on the webinar, if you have additional questions, make sure to submit them at this time. Any that we haven't gotten to during this session, one of the webinar speakers will follow up with you individually via email after the webinar in the next few days. And that last question is, if we have VCF files of our breeding parents, is this enough for you to generate our own set of markers for screening an F2 generation, for example?
What was the first part of the question? Say it again. VCF?
I'll read it again. If we have VCF files of our breeding parents, is this enough for you to generate our own set of markers?
Yeah, I mean, we normally like to, what we do, we go back and forth between us and the customer client that we work with, and we that way we ensure that we work on we work together to develop markers for that specific population. So in this case, we have to probably review that one first and then give an proposal and go from there and see if how we would develop something for that, for the traits of interest.
Okay, thank you, Asgar. Okay, and with that, we are going to wrap up the webinar. I want to remind everyone that a recording of this webinar, along with a copy of the slides, will be available to you within 2 business days. You'll receive an email. Thank you, Asgar Shir, Garrett Zielinski, and Anthony Lai for sharing your presentations and your expertise with us today. And thank you, everyone, for attending.
Thank you, Genevieve.
Thank you.